Hi guys, welcome to the third video of this session, DNA profiling. Video number 1 we have looked application of DNA profiling. Video number 2 we have looked about the non-coding region of the DNA, that's mean introns, how introns are specific and why we use introns. Now let's say two person number A and person number B. So if you take these two persons, they have this exon and introns organized like this way. So let's say that's a person B, and I'm going to show you here the exon part. That is okay. So look at here, this one is a introns, and this one is exon. As you already know that exon is coding region, introns are non-coding region. In the second video we have looked the mini satellite and the micro satellite. It's a repetitive sequence of the introns. So as a result of repetitive sequence, let's say in this person the repetitive sequence, let's look at ATC, and this ATC is repeating here probably 50 times, and the ATC here are repeating probably the 100 times. So as a result of that, the ATC regions are different from this length. So now what we have here, imagine this is the another site here. So this site is the restriction endonucleases cut site. So restriction endonucleases cut site, and then above we can also show the continuation of this DNA. So here continuation of this DNA. Alright, now restriction sites, which are called palindrome site. The palindrome site, again, they have a very special sequence like A, C, G, T, T, and here the base pairs are T, T, G, C, A, and 5 prime to 3 prime, and here 3 prime to 5 primes. You can observe here the 5 prime to 3 prime, it's A, C, G, T, T. So if you go this direction, it's the same, and when you read from again 5 prime to 3 directions, it's A, C, G, T, T. That's a palindrome site. So that's a site for the restriction endonucleases. So when you add restriction endonucleases isolated DNA, so what will happen next, exactly the process is they cut this DNA here, and they cut the DNA here. So probably I can show you like here, they cut this DNA. So when they cut this DNA, it just separates these two ends. When they separate these two ends, these sites are open. Like here now, I can show you it's A, C, G, T, T. And this site is a T here. So these sites are now, hydrogen bonds are broken in between, so they are like ready for binding with another molecule. These hydrogen bonds are really open, so I call it sticky ends for these regions. So now they cut this DNA like that way, the region. So when we cut this DNA, you can already observe if they cut in another region of the restriction site here and restriction site here, the DNA, so we can get two different pieces. This DNA size is from here to here, and this DNA is from here to here. So there are two different sizes now. By that way, when we add restriction endonucleases, we can get different fragments of DNA. So different fragments come because of the short tandem repeat. Check my video number 2, what is short tandem repeats. So these short tandem repeats are different from one person to one person. For example, we just ATC, it's a short tandem repeat. That means a trinucleotides, they repeat 50 times, and here trinucleotide here they repeat 100 times. As a result of that, these regions are different. So that's very unique from one person to one person. In USA, the CODIS system, just used for the forensic DNA analysis, and the CODIS system, they look actually 13 loci, that means 13 different locations, and check the repetitive DNA sequence. So if they look 13 different regions and this kind of a repetitive sequence, the maximum probability is a 1 in it's a power of 18th. So this kind of huge number of probabilities, that means it very unlikely the same size of bands we can observe in a person's DNA profiling. So it's very unique system because of the short tandem repeat, and they are very different from coding non-coding region of the DNA from one person to another person. It could be same for maybe a species to species. It could be same for many the similars, the family members. They are different again. The non-coding regions short tandem repeat. As a result of that, when we add the restriction endonucleases, or maybe one DNA restriction endonucleases, or maybe 2 restriction endonucleases, or maybe 3 restriction endonucleases, we get various fragments into our test tube from this person, and maybe this person I get again different size of DNA fragments. So this number 3 video, we see what exactly the DNA, how we can cut DNA into different fragments. So when you have different fragments, the last video we can go the lab technique, how we can visualize this, because DNA are very small. Even though they cut and we describe like this, we don't see anything. What is the length and what is size and how this DNA. So next step is visualizing the band, the different band patterns to check how the DNA is working.